Morning, everybody. Can you hear me?
Yes.
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 It's my pleasure to introduce Dr. Carol Blair this morning as our speaker. She's actually a native of Colorado, not Colorado, Salt Lake City. And she has graduated from University of Utah and then proceeded to go to University of California, Berkeley to get her PhD. And then she traveled all the way to Ireland to Trinity College, Dublin University, and to do her postdoctoral research. Then she came back to Colorado in 1975, and she joined CSU. And she started to do research on arthropod-borne viruses. And she continued to do this research until today. She's retired professor. She's university professor emerita in the Department of Microbiology, Immunology, and Pathology. And she's gonna talk to us on my life with viruses and mosquitoes. And here is a Carol.
All right. Thank you, Young. Let me do this share screen thing again. Can I do it this way now? Is that okay?
Yeah.
Can
Yes.
everybody see that? Okay. I first of all want to thank Young. That was, I'm very honored to be invited, and thanks to all of you for coming out to hear me. And although I'm not going to talk about coronaviruses, I will be happy to answer any of your questions about coronaviruses and bats, because I have colleagues who work on those, and I have kept up with the latest research. But what I want to talk about today is my own research on arboviruses. And so let's see. I will first of all tell you what is a virus, and I'll tell you about how I got started with arbovirus research, and I'll tell you why I think arboviruses are so fascinating, and I'll give you probably a lot more than you want to know about arboviruses and mosquitoes. But in the last few years, we've been trying to prevent or see how we could control arbovirus diseases through manipulation of the mosquito immune system. So that's what I'm going to finish up with. And please feel free to stop me with questions at any time, although I can't see the chat room. I think Marisa is going to tell me if there's a question, right?
Correct. Yeah, keep an eye on the chat, and if anyone wants to unmute, you're welcome to do that too and ask a question.
Okay. All right, so first of all, as you probably know, viruses are very small. They are too small to be seen in a light microscope the way we can see bacteria. So here is a log scale of size, and you can see bacteria are about 1 micrometer average. Viruses are about 10 times smaller than those, so they can't be seen in the light microscope. They can only be seen in the electron microscope. I'll show you, whoops, there. Some beautiful electron micrographs. I didn't take any of these. of viruses in the electron microscope. This is a bacteriophage, a virus that infects bacteria. This is tobacco mosaic virus. It infects plants. This is a rhabdovirus. It infects horses, cows, and humans. And this is a rotavirus. And these are all about on the same scale. And so you can see rotaviruses are very small, and they cause serious diarrhea in infants. So there's a good vaccine out for rotaviruses now. Okay, briefly, this is how viruses multiply. They can't multiply outside a cell. So they have to get into a cell and then use the machinery of the cell to make new parts to assemble into new viruses. So first of all, and I don't know, can you see my pointer? No? Okay, anyway, number one, there are receptors on the outside of the cell that the virus attaches to. So you've heard about the ACE2 receptor for SARS coronavirus 2. There is a very specific receptor on the outside of the cell for any particular virus. Then the virus enters the cell, usually by a bulge inward on the cell membrane. And then after it gets inside the cell, number 3, it falls apart. So the virus just disintegrates, and it allows its nucleic acid, and I'm going to talk about RNA viruses. The nucleic acid then gets into the cell, and it is both made into new proteins, and then it is replicated. So you've got a whole bunch of new proteins, you've got a whole bunch of new virus genomes, and then number 6, they assemble, get back together into new virus particles, and get out of the cell either by breaking open the cell or by a bulge out of the membrane in the other direction. And so how did I get started with arboviruses? I grew up in rural Utah. I love to go out in nature. I caught toads, lizards, snakes. My dad liked me to bring those back, turn them loose in his garden so they could eat insects. And I loved going hiking in the mountains. We lived right at the base of the mountains. So I just loved getting out in nature. It took me a while to connect my love of nature with actual academic science, but I had a wonderful high school chemistry teacher who just made me love science. So I started at the University of Utah as a chemistry major, and that lasted for 1 year, and I got very good grades in chemistry. But then I talked to friends who were majoring in microbiology, and I switched to a microbiology major because it had all the precision of chemistry, but all the fascination of biology. And what's more, I got a job in the lab of 2 graduate students who were studying arbovirology. They were wonderful mentors and role models, and that's how it all started. This is very detailed, but I'll just point out at the top that when I was interviewed for the job as a lab assistant, the department head asked me, "Are you afraid of snakes?" And I said, "Why, no, I'm quite good at catching snakes." And I got the job. Then I took the job. I made cell cultures. I made cell culture medium. I took care of the mosquito colony. It was Culex tarsalis mosquitoes. And I went out to catch snakes in the wild. I brought them back, took care of them, included feeding them. I helped John bleed the snakes, and we tested their blood for Western equine encephalitis virus, which is an arbovirus. And that position was a major influence on helping me decide what I wanted to do with the rest of my life. Okay, here are my snakes. I wrote my honors thesis on snakes as a possible winter reservoir for Western equine encephalitis virus. And just to prove that it would be possible, we allowed the mosquitoes to feed on snakes. And see, these are mosquitoes sitting on the head of a snake. You may not be able to see this whole one, but you can see this mosquito is full of blood that it took from the snake. And the snakes have scales, of course. The mosquitoes just push their proboscis in through the under the scales to get out the skin. And this is a photograph of a different kind of virus, a different kind of mosquito. It's called Aedes aegypti. These are the mosquitoes that I've been studying for the last 40 years. So arthropod-borne viruses, shortened to arbovirus. And they're a large, very diverse group of viruses, and they are transmitted to vertebrates, including humans, by blood-sucking insects. The official word is hematophagus. And the really fascinating thing to me is that the virus replicates in the mosquito, and so it expands in the mosquito before it is transmitted when the mosquito is taking blood out of its human host. And these are natural transmission cycles. So most arboviruses, their natural vertebrate host is not a human, it's a horse or a bird or a rodent. There are a few, like dengue virus, that their transmission cycle only involves humans. But in most cases, humans get infected when the virus spills over from its natural cycle into humans. And this is like the center one is like West Nile virus, where its natural host is birds, but those mosquitoes can also bite humans and transmit the virus to humans. Now, what is so fascinating to me about arboviruses is that they do replicate very efficiently in both mosquitoes and humans, and those are physiologically very different hosts. So the strange thing is that arboviruses cause cytopathic infections. That is, they can cause serious diseases in humans. But in their mosquito host, they do not cause disease. They do not cause cellular damage, and they persist for the life of the mosquito. So the mosquito might live 3 weeks, and during that entire period, she is able to transmit the virus every time she bites another bird or human. So my hypothesis from a long time ago was that the different outcomes of arbovirus infections in humans and in mosquitoes was because of the differences in their host immune responses. And I don't have a slide about this, but I'll mention, and those of you, particularly if you have been vaccinated against COVID-19, you are aware that as the human immune response kicks in, it can make you feel pretty bad. And in fact, you're lucky to be vaccinated, because it has been found that the SARS coronavirus 2 can cause serious immunopathology. So the very serious cases of COVID-19 involve an over response of the immune system.
I have a question.
Hey,
Do male mosquitoes, they feed on plants, do they not get the virus because they don't consume blood?
that's right. Male mosquitoes are not infected by arboviruses. There are some other viruses that infect only mosquitoes, and they can infect both male and female. But arboviruses only infect the female because they only, those are the only ones that take blood from infected vertebrates. So this is the mosquito cycle. Here's the number 4 is the adult female mosquito. She takes a blood meal, and then only after she has taken a blood meal is she able to lay eggs. Of course, she has to have mated with a male mosquito, but that happens all the time. The males are really persistent. But anyway, after she has mated and then had a blood meal, she can lay eggs, and you can see the eggs are really tiny. This is a pencil point, and each of these little dots is an egg or a huevo. Here's a magnified version of the eggs. Then these eggs hatch and they become larvae, and these larvae have to swim in the water. Then the larvae eat and they develop, but then they become these shelled creatures called pupae, and the pupae don't eat. They just develop further and then they hatch. And right down here in number 4 is a pupa and one that is just hatching into a mosquito. So that's the cycle. We carry that out in the lab. Here are some Aedes aegypti. The eggs, which have been laid on a paper towel, the larvae, the pupae, and the adults. Okay, here's how the mosquito gets infected. The female mosquito takes a blood meal, and here's a diagram. It has its proboscis pushed down in through your skin into your capillary. It pulls up blood. This red line is blood coming from your capillary up through the mosquito's proboscis, down through its alimentary tract, into its posterior midgut. And these little circles represent viruses. The virus then infects the cells in the posterior midgut. It multiplies there. Then it's released into this, which is called the hemocoel. It's a big empty space in the mosquito's thorax. And it infects various organs, including the salivary gland. So this is the salivary gland. And it replicates there, and then next time the mosquito takes a blood meal, it goes out with the mosquito saliva down into the capillary. So that's the whole cycle. Depending on the virus, this can take various amounts of time. The dengue viruses that I work with, depending on the temperature, take 10 to 14 days to go through this whole cycle. Okay, now can you all see the full slide?
Yes.
Okay, because I'm all right. So this is how we measure the development of the virus in the mosquito. This was done by one of my favorite colleagues, Irma Sanchez Vargas. We infect the mosquitoes with an artificial blood meal, and then the amount of virus in the mosquito drops for a few days when the virus is beginning to replicate. Then in the midgut, this red line it goes up until about 10 days. It reaches a maximum, but then it goes down again. But after it escapes the midgut, the titer of virus in the mosquito carcass, which includes the salivary gland, just stays at a high level for 14 days or more. And these are immunofluorescent images of the virus in, first of all, the salivary gland. Here's the midgut and salivary gland at 10 days. Then after 10 days, there's no or very little virus in the midgut, but the salivary glands just continue to have a lot of virus. Okay, so dengue, the virus I've studied most in my career, can cause very serious disease in humans. And there are probably 400 million cases of dengue virus throughout the world, mostly in tropical regions, each year, that many. And 100 million of those develop disease. So it would be important to prevent that disease. Now, there are several possibilities. We could develop an antiviral therapy, but so far there is no specific treatment for dengue virus or for most other RNA viruses. We could develop a vaccine. That's been going on for 50 years, and there still is none that is both effective and safe. And so what we've concentrated on is mosquito control. And we've looked for new ways because mosquito control has usually involved insecticides. And the mosquitoes develop resistance, and insecticides can be very bad for the environment, as we know. So we're looking for new ways that don't involve insecticides. And so we're looking at how do mosquitoes handle virus infections? What is their immune response? And is it possible that we could manipulate the mosquito immune response to block virus transmission? Okay, this is complex and I won't dwell on it because this is the mosquito immune response, but it was first discovered and studied in Drosophila. Now, Drosophila are fruit flies, but they're not too far on the evolutionary scale from mosquitoes. So we have looked at what was learned in Drosophila and the siRNA pathway, a pathway of RNA interference is the one that mosquitoes use to control arboviruses. So when the complete genome of Aedes aegypti was first published in 2007, it's huge. It's a huge genome. But we were able to find the genes in the mosquito genome that are equivalent to those in Drosophila for antiviral immunity. And this is how it works. This in number one, this is double-stranded RNA that occurs as the virus replicates. And there are several mosquito proteins, dicer, R2D2, and several others that bind to this RNA. They chop it up into little pieces, and then they deliver it to another protein that's called Argonaute 2. And this green ball is Argonaute 2. It takes a little piece of RNA and uses it to guide itself to the to the arbovirus genome, and it cuts the arbovirus genome right here in the center of where the little piece of RNA bound. So the virus RNA is chopped up inside the cell. And we found that mosquitoes have all the major components of RNA interference, and that it plays a major role in mosquito antiviral immunity. We demonstrated that if we block those dicer 2 or argonaut 2, then the virus kind of goes wild in the mosquito. So, whoops, we wanted to know, could we, now, there is a balance. The mosquito doesn't quite eliminate the virus. Unlike the human immune system, where the virus is eliminated, the mosquito is not able to quite eliminate the virus, but it tamps it down so that the virus doesn't kill the mosquito. And the virus doesn't want to kill a mosquito because it does want to be transmitted by the mosquito. So it's a kind of balance between the virus and the mosquito. Now, we said, could we ramp up the mosquito response to arbovirus infection so that we could completely prevent the virus from being transmitted? And I promised Ah Young I wouldn't give you a lot of data, but this is a diagram of an experiment that I have to attribute to our former colleague, Alexander Franz. He's now a faculty member at the University of Missouri. He injected mosquito eggs. Remember how small the mosquito eggs are? He is a champion at injecting mosquito eggs. So he injected them with this piece of DNA, which incorporated itself into the mosquito genome, and it caused a very rapid RNA interference response as soon as the mosquito took a blood meal. And so here's the only data I'm going to show you. This is the midgut of a mosquito that didn't get the DNA inserted in its genome and was infected with dengue virus. This is a mosquito that did get the DNA into its genome, and now it's resistant to infection. And if we determine how much infectious virus is in each mosquito, each of these little squares in the diagram on the right represents the amount of virus in one mosquito. And so the red ones are the non-genetically engineered mosquitoes. The green ones are the genetically engineered mosquitoes. And you can see at 7 days, at 10 days, and even at 14 days, these mosquitoes are virtually making no virus. They're not making enough virus that it can be expelled into the hemocoel and get to the salivary glands. So those mosquitoes simply cannot transmit the virus. So we did find that we can manipulate the genome of the mosquito to make it completely resistant to dengue virus. So I'm going to wrap up here and then let you ask me questions. But our overall summary is that arboviruses cause persistent infections of mosquitoes, and that the mosquitoes can transmit the virus to humans throughout their lifetime. And the mosquito antiviral defense is called RNA interference. And arboviruses don't want to completely evade RNAi. They just want the mosquito to replicate enough that they can be transmitted. But we can genetically manipulate mosquitoes so that they are completely resistant to arbovirus infection. Okay, here are some of my colleagues. I've worked with wonderful people all my life, still working with Ken Olson up here at the top with the beard. And here are some of their names. On the left are faculty members. On the right are some of my great graduate students. And our research has been funded by the National Institutes of Health. Okay, and I am going to leave it at that and then let you ask me questions if you'd like to, or make comments or whatever.
I have a question.
Okay,
My daughter got dengue, and
really, where was she?
well, she works in Bali, and she
Ah,
got it while she was on the beach, just before she came home, and so
mhm.
she didn't have it till she got here, and they said, "Oh no, you have malaria," and
Yeah.
she said, "No, I think I have dengue," and so they made her pay for all the malaria tests, and then she didn't have it, so
Uh-huh.
they decided then they'd test her for dengue, and lo and behold, she had it. But I've read that there are 4 major strains of dengue, and if you get one, you can get the others, and they will be more serious. With each reinfection, it'll be more serious. Is this true?
Absolutely. You've been reading the right things. Yeah. That's one of the reasons that it would be so important to have a vaccine for dengue. But you would have to block all 4 of the serotypes with a single vaccine in order to prevent that more serious infection by a second serotype. But yes, absolutely. Was she very sick?
She wasn't very sick, no, but she works in Bali, and so
Mhm.
she's going to be exposed more. I mean, she came home, fortunately. Her visa expired in July, so she's been home and working from home, but she's going back there. So I
Okay.
always worry about her.
Yeah, and it is so hard to avoid mosquitoes in the tropics because they're everywhere, and Aedes aegypti bite during the day, unlike the culex that live around here.
And they live in houses, don't they? I
Oh,
mean,
they love houses, yes,
mhm.
because their main source of nourishment is human blood. I'm sorry to give you that bad news, but you've been reading the right things, so you already know that.
Carol, have you traveled quite a bit doing this research, or have you been more just here in the States doing it?
I have not traveled to actually do field research, but I have traveled to some really interesting places to meet up with other arbovirologists, to give talks at meetings, that sort of thing. One of my favorite places was Cuba. And I shouldn't talk politics, but I'm hoping with the new administration we'll be able to go back to Cuba because they have done very good dengue research.
Hi.
Hi, Amy.
Hi, Carol. My mom, Ardith Kirst, is a member of the CSU Women's Group, and she forwarded this invitation to me. So I'm so delighted to get to join by way of invitation. I'm one of Carol's former students and
Yes.
current colleagues. So this is just a pleasure. Students are a lot like children. They think their relationship with their professor is all about them. And
And it
so,
is.
so it's such a pleasure to get to hear this nice talk about your really life's work with
Yeah.
vector-based efforts for control and prevention of arboviral disease. I
Yeah.
have a kind of forward-facing question. What do you think the role really functionally can be for genetically modified mosquitoes in public health and control and prevention of orphan, particularly arboviral diseases where we don't have viable vaccine alternatives? And I'll just
Right.
leave it there. Thank you for the lovely talk.
Well, thank you. And let me say that Amy is now, she has a very responsible position at the CDC that's just across the road from where I am now. And she was a great graduate student and still is a great scientist. So that is one of the important questions that has grown out of our research, because in order to release those genetically modified mosquitoes, you have to jump through lots of hoops, as you might imagine. It is not easy to anticipate all of the problems that might arise from release of genetically modified organisms, and especially mosquitoes that can go everywhere. So we haven't pursued that kind of modification. What we're pursuing now is to introduce either bacteria or viruses into the mosquitoes that will release double-stranded RNA that will block the replication. And that is more acceptable because bacteria and viruses naturally live in mosquitoes. So you're just tweaking the organisms that naturally live within the mosquitoes. So that's, but we still think it's a very good way to control transmission of arboviruses. And you've probably read about Wolbachia, so I won't go into that, but that is a bacterium that naturally infects mosquitoes that can reduce virus replication.
I have a question. Do you think there will come a day when the mosquito will be able to replicate the coronavirus and transmit it to us?
I don't. That's not an absolute, but you know, when SARS broke out in about 2003, it was, of course, tested, as was Ebola and all of the other viruses that emerge in tropical regions, to determine whether or not they can be transmitted by mosquitoes. And there was no evidence that coronavirus could infect mosquitoes. Yes, I have a colleague, Dick Bowen, who he is a veterinarian and loves working with animals. And he has done much research on all of these emerging viruses, including the first SARS virus and the MERS virus, which is also a coronavirus. And one of his graduate students got in touch with me one day and said, I can't make it to class because I have to collect nasal swabs from camels. So yeah, I work with a lot of fun people.
Yeah. Hi. Can you hear me?
Yes.
Yes. Okay. So now in the beginning, you said there is no vaccine available for this virus.
Mm-hmm.
And can we not? Well, we have right now a vaccine available for coronavirus and so on, using biochemical technique, okay, putting in RNA and so on. Since it is an RNA virus, can we not use the similar technique to be able to do the vaccine? I am surprised to hear that you haven't had it for 50 years or something. Can we?
Well, the use of RNA as a vaccine is quite new. It's been in development for 10 years, but these are actually the first vaccines that have been approved for human use
Yes.
that are RNA vaccines. But I'm thinking that this breakthrough might help to develop an effective dengue vaccine.
Uh-huh.
There was one developed a few years ago, and we heard about it over and over again at scientific meetings about how good it was. And so it was actually licensed in the Philippines and in Mexico and in several other places. And they vaccinated a huge number of children in the Philippines. And then it turned out that some of those who had not been infected with dengue before and got the vaccine, when they did get infected with dengue, developed a much more serious disease. Like the earlier questioner was asking, if you get infected a second time with a different type of dengue virus, you can get very seriously infected. So that vaccine is not used anymore because it caused such serious disease in some children.
I see. Thank
But
you.
yeah, we
Yeah.
hope for messenger RNA vaccines.
Yeah, right.
So, Carol, where are things in terms of the approach of using bacteria and whatever else to inoculate, I guess, the mosquito? Since that approach seems to be a better approach than.
It is, and we've done a little research on it, but we ran out of funds before we developed the bacteria. There have been some successes in preventing malaria. Well, again, experimental. They haven't been released into the field, but with bacteria releasing an anti-malarial protein that blocked the development of the malaria parasite in the mosquitoes. Those are a different kind of mosquitoes. They're anopheles, but there are a lot of people working on it. So
Yeah, because that reminded me, yeah, Bill Gates,
Mhm.
Bill and Melinda Gates and their foundation, they have really zeroed in on malaria, I think.
They have, yeah.
So they would have money and maybe there'd be a crossover from
Right.
their research into other areas.
Into viruses, right.
Well, maybe things are looking up.
Yeah,
You know, I like to be optimistic, so.
right.
Linda, you're on mute. I think you're talking.
Oh,
I have another question. I don't know who Linda.
okay.
Okay,
Oh.
go ahead, Mary
Can
Jane.
Can one mosquito carry more than one of the strains of dengue?
That's a good question, and probably yes. Some of my colleagues have shown that one mosquito can carry both dengue virus and chikungunya virus. But I think they could carry two strains of dengue virus. And the reason I think that is that we have shown that if you induce RNA interference against one serotype of dengue, it's not effective against the others. Linda, you're on mute.
Unmute. Okay,
There you
No.
go.
got it now. Okay, is West Nile virus, which is a common in our area,
Mhm.
related to all of this or any of it?
Oh, yes. Yeah. West Nile virus is in the same family as dengue virus, but it's transmitted by Culex mosquitoes, not by Aedes mosquitoes. And surprisingly, although those are very closely related, they have quite different immune responses to viruses.
Is anyone working on that in this at CSU?
Yes, some of my colleagues do a lot of work on West Nile virus, but not specifically on trying to develop resistance to in mosquitoes. So it's more, well, it's more about the ecology and the evolution of West Nile virus. But yes, we haven't done anything like that on West Nile.
Interesting.
And I think there are some good therapies for West Nile virus, but it's very hard to do a clinical trial on the therapies because you can't predict when West Nile virus is going to break out again.
Mm.
We have a few cases every year, but no epidemic.
Right.
Carol, what
Thank you.
about Lyme disease? What about Lyme disease? How is
Good
that
question.
transmitted?
It's transmitted by ticks, and
Right.
it is caused by a bacterium. So it's a bacterial disease.
Oh, that's right. Yes.
That's transmitted by ticks. Yeah.
Mm,
I think
yeah.
Amy would know about research on Lyme disease at the CDC.
Yeah.
I
Thank
can
you.
say in general, Lyme disease is so politicized that one of the huge challenges within the agency is to try to help the public separate fact from fiction and justify kind of intervention-based methods based on fact when there's a lot of disinformation out there. So I would encourage anyone with specific questions or needs about Lyme disease to reach out to the Centers for Disease Control through their website.
Yeah, the CDC has wonderful web pages on most of these diseases. Yeah.
You know, it's kind of interesting. I know somebody who got Lyme disease, but got it in visiting a daughter in Massachusetts.
Yes.
And then a friend here who's moved to Denver was a nurse who got the West Nile virus. And they stay with you a long time.
Oh, West Nile
They're-
can.
Yeah. Yeah,
It can
Joan
cause
yeah.
nerve damage that lasts forever. Yeah.
So what are we finding out about the coronavirus, and do we know anything about long-term effects of that?
Well, we haven't
Had it that long?
We've only got a year's experience, but yes, it does seem that some people have very long-lasting effects.
Do you have any idea of how this might end? Herd immunity? I mean, have there been illnesses in the past that just were similar and just faded away?
We really have to get the vaccine out to as many people as possible in as short a time possible, because as we have seen recently, new variants of SARS coronavirus 2 are emerging, and the one in South Africa is quite frightening because it does not respond as well to the vaccine as the original coronavirus. So to get people vaccinated so that they will have that variant won't have a chance to spread and further mutate. The vaccine does give some protection against that variant.
So vaccinations and time through.
And wear your mask and social distancing.
Right. Okay. I feel empowered now that I've had both of my vaccinations.
Yes, I do too.
But still a little bit fearful. So good. We'll keep up the good work. Thank you, Carol, so much. What a fascinating and so topical subject. We thank you for the research you've done all of these years. So excited that you were interested in playing with snakes and creatures and that no one said to you, ooh, girls don't do that.
Oh, you bet they do. And I raised baby bats as well.
Really? We need more of this, do we?
Yep. Well, thank you. It's been an honor, and it's been fun talking to all of you.
Thank you.
Thank you very much.
Thank you.